Hi everyone, it is time for Tissues Part 2. We are going to now hit our connective tissue content for our Bio 207-695 hybrid course. So we've talked about epithelial tissue, and remember epithelial tissue is two parts. There's the glandular epithelium, which is going to form endocrine and exocrine glands, and the exocrine glands make secretions like sweat, saliva, your sebum, your oil, your earwax glands, and then your endocrine components are going to put hormones into the bloodstream. And we'll talk more about that at the end of the semester when we get to the hormone component of the class. The other side of the epithelial tissue is the protective component, where a uniform-looking cell, so all of the cells are squamous, all of the cells are cuboidal, all of the cells are columnar, all of the cells have this uniform look, and they're either single, simply one layer stacked, or they are multi-layered, and so multiple layers are stacked. But the common unit is the cell is not unique. It's the same repeating kind of cellular pattern. They just happen to be touching each other. When we start talking about connective tissue, we're going to find that there are multiple potential cells located within the tissue. So you might have a immune cell next to a resident connective tissue cell. And those two cells are going to be extremely different, not only in what they look like, but in what they do. The resident fibrocyte cell is there to make proteins that are part of the background matrix material, whereas the immune cell might be there to help should something get past the epithelial protective layer, the connective tissue that's the glue of that basement membrane, helping that epithelial tissue stay bound to the body and to underlying tissue, is going to sit there, and should some pathogen get past those barrier cells, be available to attack it or handle killing, trapping, and destroying it. So when we look at epithelial versus connective tissue, we see that connective tissue is going to vary largely in appearance, because we're not looking at cells touching uniform-looking cells. We're looking at, there's a lot of background material, and that background material might be dense with a lot of proteins that are fibers that are large-shaped proteins that you are going to see arranged in a loose pattern, a thick pattern, or a arranged orderly pattern. We might see that the background material is mostly fluid, mostly water. We might see the background material is a calcified, crystallized, hard structure. So the matrix, the background non-living material, can be a variety of different things, and cells suspended and living in that background material are going to be multiple potential cells, not just a uniform single cell, but a multitude of potential cells can live in that environment. All right? And so that is the big difference of connective tissue versus epithelial tissue. So looking at the slides, when you look for epithelial tissue, you want to see a repetitive cell with a nucleus over and over and over, touching, one side exposed, one side connected to underlying basement membrane, some of those cells undergoing mitosis. When you look at connective tissue, you're going to see cells, and you'll know they're cells because they'll have their nuclei with their DNA, RNA staining a little darker, and you're going to see them kind of embedded in this material, and that material might be fluid, like in blood. It might be like a ligament or tendon, a very dense, rich protein population. It might be in bone, a crystal carbonate structure. It might be in cartilage, a jelly-like substance. But the end result is connective tissue. There are cells, immune cells, chondrocytes, osteocytes, mesenchymal cells, a multitude of potential cells that can live in a non-living suspended background. Right? And so depending upon this type of background, depending upon the cell population, this connective tissue might just be a glue-type material that helps connect epithelial tissue to the rest of the body. It might be it is a tissue that connects connective tissue to muscle, so bone to muscle, bone to bone. It could be the bone itself that is wrapping around, like the pelvis or the skull or the ribs, around other tissues underneath and other organs. It's going to be a little bit of our fat, our kind of cushions. It's going to kind of take up space where we have overlapping organs or overlapping, material of an organ. It's going to store cells that can store, like fat cells, large amounts of triglycerides, which are basically fatty acids for energy consumption that are stored in these molecules and then stored largely in the cell. All right, and the immune system falls into this category, so a function of your connective tissue and focusing on the immune cells means this is a way to help protect the body from harmful invaders. Okay, so we know connective tissues as a cohesive unit have a non-living background material, and cells live suspended in that background material. Cells can be of a variety in nature. When we look at the matrix, the extracellular matrix, this non-living background material, what it's composed of is going to be a large part of how we then categorize that tissue. So connective tissue proper is going to be connective tissue that has a matrix that is of a certain protein content, a certain background material content that helps connect items to each other, like ligaments and tendons, dermis tissue that connects the epithelial epidermis layer to the underlying hypodermis and then muscle layer. And because it's found where maybe glue is needed to hold things together, it's going to help protect the tissues by maybe holding the epithelial tissue to the underlying muscle and the underlying body. Okay? Fluid connective tissues are your lymphatic and your blood, and fluids move through tubes, and as they move, help things move from one area of the body to another. So they play a big role in transport. Most of the items being transported, because the matrix is water, the item needs to be suspended and dissolved in the water. So for triglycerides, for steroid hormones, for cholesterol, in many cases, in order to carry those items through the blood, through the lymphatics, they have to be bound to proteins that make them water soluble. Supporting connective tissues are going to be our bone and our cartilage, which it's a connective tissue because cells are suspended in a non-living matrix, but the non-living matrix has a little more robustness to it that basically provides a framework for the entire human body to then build off of. When we start looking at the categories of connective tissue proper, all right, you're going to look at, is the connective tissue a spider web, thin, somewhat small or loosey-goosey type tissue, and if it is, then it falls under the category of loose connective tissue. So this tissue can somewhat be a little bit of structural protein fibers that are providing a little bit of a framework, kind of like a spider web's threads, that then other items can flow into, like blood vessels, like nerves. And so it's enough of a support and glue to kind of hold things together, but it's still open enough that passageways for vessels, for neurons, for other items to move through can occur. Okay. Examples of loose connective tissue are your fat tissue, also your areolar connective tissue is here. Okay. When we need the connective tissue to help bind bone to bone, or help bind bone to muscle, or help connect the epidermis to the underlying muscle, we are going to see that we need a more robust matrix type material. And so what we're going to see is that the matrix is going to be less open and more of like weaving of fibers or knitting of fibers together. And by knitting the fibers in a very tight knot together, you'll end up with a very tough, strong matrix that cells live within, and that matrix has some ability to withstand lots of forces being pulled upon them, created by maybe muscles that are interconnected to it. And so that's where tendons and ligaments can come into play. So the matrix is important. What is the background material? And that background material doesn't just randomly snap our fingers appear. That background material is laid down and put into that environment because of the cells that make it and then exocytose it or secrete it into the environment. So with the variety of background materials we have and the background matrix being so diverse, we know that many different cells are going to potentially be involved in providing the uniqueness that each individual connective tissue has. For most of our connective tissue proper, so our loose connective tissues and our dense connective tissues, we know that there is going to be a population of these structural supporting proteins. And these structural supporting proteins are much like your knitting yarn. They are knotted, they are laid down in a pattern and a manner that allows the tissue to at times be very strong, at times be very bendable or elastic. And so that the protein characteristics, whether it's a strong, tensily strong protein or a elastic protein or a protein that provides a lot of openness and looseness, is going to come into play when we name it. These proteins all come from genetic expression of certain genes. So embedded in every cell in our body, we have genes, and the genes for collagen, elastin, and reticular fibers are embedded in every cell. But it's only certain cells that express these genes. And uniformly, the cells that express these genes are cells that reside suspended in matrix of connective tissue proper, and they are called fibrocytes. And if the fibrocytes are building matrix, we tend to call them fibroblasts. So a building cell of the matrix is a fibroblast. A resident cell that might be inactive at the time might just be called a fibrocyte. So when we look at all of the connective tissues that are proper and loose, you are expecting to find, if there's collagen and elastin and reticular fibers, if there's those proteins present in that background material, there has to be somewhere some resident fibrocyte or fibroblast cell that is going to go through transcription, translation, exocytosis, and put those proteins in that extracellular matrix. Okay? Now, another form of loose connective tissue proper is fat tissue, adipose tissue. And the cells that reside in adipose tissue sit in an extracellular matrix that is a little bit of reticular, collagen, and elastin fibers. But the main cell of this tissue is known as an adipocyte, because this cell, yes, it can make collagen, it can make elastin, it can make those proteins and put it in the ground and the material around it. But the main focus of these cells is to hoard the body's triglycerides, the stored energy molecules that the body keeps for later on when fuel availability is low because of maybe diet or maybe because of exercise or just the calories coming in or not meeting the demand of calories expended. From a developmental point, all of your cells for fibroblasts, fibrocytes, adipocytes are derived from a common unit, a common cell. And when you are growing, some of the cells haven't finished developing, haven't finished being differentiated into fibroblasts, fibrocytes, or adipocytes. And they are in some ways the stem cell that still has the ability to become what will be a fibroblast, become what will be a adipocyte. And so in that kind of step or two up from being terminally differentiated, we call those cells mesenchymal cells. when you get older, so when you start to be my age and older, your population of mesenchymal cells is very low. It's depleted. And so that's part of the reason why I'm going to be prone to get wrinkles or have longer healing of any damage to connective tissues and more likely to scar if I get cuts and bumps, because the population of stem cells that can become collagen, elastin, reticular fiber producing cells in my dermal tissue of my skin or in my tendons and ligaments of my body are just a smaller population because I'm older and I've used a bunch of them as I've grown older. Okay? So the mesenchymal cells are in some ways those original stem cells, resident cells that still have somewhat an ability. If you need to make more storage cells for fat because you take in a lot of calories and you have oversupply of fuel, you will push some of them to become more adipocytes. If you have, again, damage or cuts and bruises and scrapes and you need to redo and add to your fibroblast and fibrocyte population in your connective tissue of your skin, the mesenchymal cells can do that for you. Okay? All of the other cells listed here, except for melanocytes, are immune cells. They come from your bone marrow, and they are going to hitch a ride in the blood supply and eventually make their way behind every epithelial barrier of your body, and they sit behind those epithelial barriers in the event that a virus, a bacteria, a fungus, a worm, a termite, a little mite, a little flea was to get past that epithelial lining. And they have different roles in immune physiology of how they work. Some of them eat the pathogen, some of them spit on the pathogen, some of them pull other cells towards that pathogen to help out with breaking down, killing, and eating. Okay? Melanocytes are going to be pigmenting cells. So melanocytes are going to be cells that technically reside in the dermis of the skin. They reside in areas of the eye. And what they do is they produce the protein known as melanin. And melanin, we know, is what gives our skin some of the coloring that we associate with skin color. Melanin helps to absorb ultraviolet radiation and helps protect our skin from cells being damaged and their DNA getting broken and potentially mutated and the cell becoming cancerous. So melanin is going to be embedded near the epidermis-dermis layer, and as the cells are stimulated by high levels of UV radiation, you'll see they'll make more melanin, and that melanin will be distributed to the epidermis cells to help them, along with keratin, be waterproof, but also absorb more ultraviolet energy before it gets to the deeper layers of the skin where DNA and active mitosis and DNA gene expression is occurring, which could be damaged, disrupted, or ruined. Okay? So when we think about connective tissue, if we talk about connective tissue of the skin, connective tissue of the mouth, connective tissue of the heart, connective tissue of the kidneys, connective tissue of the of any organ of the body, we have a potential for that area to increase, decrease its collagen, elastin, reticular protein populations, and it can become a tissue that's stronger or more elastic, or with time, less strong, less elastic. We also know that there will be immune cells in those tissues, so anytime we have immune cells, there's an ability of that tissue to potentially undergo an immune response and get inflamed and end up with some type of itis, right? So you end up with some type of inflammation response in an organ or in a specific tissue. Okay? I've mentioned these three proteins. The three main proteins we look for in connective tissue matrix are, for strength purposes, collagen fibers. That's what gives connective tissue some ability to withstand forces being applied to it. Reticular fibers are the fibers that help us, kind of like in the Eiffel Tower. They are the steel beams that help kind of interconnect and give structure, but because of their arrangement, they allow for a lot of open space. So fluid could flow through, or big blood vessels could flow through, or organs could potentially kind of push into big open spaces in that area. Elastic fibers are like elastic rubber bands. They have a lot of recoil and stretch to them, and they are what allows our organs that need to be stretched or contracted, the background material that muscles are pulling on, to have that ability to be pulled and then pull the muscle back into a resting tension. Okay. How much of the fibers are present is going to give us an idea of where and what this connective tissue is going to do, and somewhat what its name is going to be. All right? So when we look again at the extracellular matrix of connective tissue, we expect there to be not one uniform cell, but lots of variety of cells. We expect there to be an openness, and the openness will typically be filled with watery fluid in the background. All right? If there are a lot of proteins arranged densely, we will see less water. We might see the cells are squished in between those proteins. If we see more loose and openness, we're probably looking at more of a looser connective tissue. So looking at this picture, it kind of summarizes why we want some of the looseness. Some of the looseness is so we can maybe pack in blood vessels, nerves, and a variety of these cells. We see that the proteins providing the structure are going to come in a lot of different sizes and shapes. So collagen fibers are tending to be very strong fibers, and so they might be more robust, a little bit more round, a little bit more of a like a chain link fence. If you pull on them in the right way, they give you a lot of ability to be strong. Elastin fibers are going to maybe be a little thinner, but they are going to have a lot more of stretchableness to them as well as recoil with them. And with the reticular fibers, which we see up here in the corner in the brown, they are somewhat like bridge wires, like the Eiffel Tower. They form a beaming kind of structural motif that is very strong but has a lot of openness to it. So should a blood vessel or a nerve or something need to be run through this material, it has space to do that. Right? Over here in this picture, it just shows you a slide of if you were to look at this through the microscope, you see, again, most of the time you see just the nuclei of the cells, but they come in a variety of shapes, so you know that the cells are a little different, and they're not the same looking cells like we saw in epithelial tissue. We can kind of see the elastic and the collagen fibers are present, collagen being a little thicker with this stain. Okay? So why do we have loose connecting tissues? The loose connecting tissue is going to be a lot of times a gluey, packagey, sticky material between other tissues. The adult human falls into 3 types of loose connective tissue. Areolar connective tissue, there's a good mix of collagen, elastin, and reticular fibers, all being produced by fibroblasts and fibrocytes. And there are a variety of the immune cells there. This is going to be the tissue we tend to think of as part of our basement membranes behind many of our epithelial tissues. So when you talk about the basement membrane of a gland, it's in many cases a little bit of areolar connective tissue. When you talk about the basement membrane in the cuboidal cells of the nephron, it's going to be that areolar connective tissue. So, and sometimes it's also known as the lamina propria when it's connecting behind epithelial tissue. And so again, anytime you hear about a membrane and it's the mesothelium, the simple squamous epithelium, and a lamina propria, the lamina propria in many cases is this areolar connective tissue. Adipose tissue, you will see adipose cells in areolar tissue, but once you start seeing a larger number of adipose cells connecting to each other, and then again, depending upon the amount of fat stored, the cells can get really, really, really large and start to take up a lot of space. You're going to be looking at large numbers of fat cells congregating together is an adipose tissue. There's still space because things can push through in between the cells. So unlike epithelial tissue where the cells are kind of tightly woven into each other, the adipose are touching and maybe largely because of size cohabitating in a general area together, but it is pretty easy for something to build in between the cells or push in between the cells if necessary. And then our reticular tissue, we're going to see again like in the spleen, like in the liver, where we want more openness, but we want just enough structure to keep the organ intact, a large amount of reticular tissue. Okay, so characteristics of areolar tissue is it's a least specialized tissue, so it's kind of a good number of collagen fibers, a good number of elastin fibers, a little bit of reticular fibers. So not any particular fiber takes a higher ratio or a higher number of content or space. It's very open. There's a very kind of gaggle organizement of how these proteins might kind of lay across each other. So it would be like if you took your Lincoln Logs out of a box and just dumped them on the floor, or you took like the whole pixie sticks or whatever, pickup sticks, and you kind of dump them on the floor. There's some openness as the sticks kind of roll around in the background material, and they can kind of bend and touch and cross across each other, but there's no really organization to it. Okay. A viscous ground substance means there is going to be some water. Some water is going to have other proteins suspended in it, other ions, and it might be a little thicker than blood and a little thicker than other areas of our body, so it makes it a little more viscous. Okay. The reason why we want the elastin and collagen and all of the reticular fibers is areolar tissue is where many of our blood vessels and capillaries are going to be located, and they need a lot of space, and they need to be able to get close to epithelial linings. And so that's part of the reason why we're going to find it in many cases as the lamina propria, the material directly behind many of your epithelial tissues. All right. The adipose tissue, again, we're going to see that the adipose cells are known as adipocytes, and they can, like our picture shows here, have a lot of background material. They might have a little bit of space in between each other. They can take a variety of shapes and sizes depending upon how much triglyceride, how much fat the body needs to store. So in people that have a high adipose or high fat percentage of their body, it might be that those cells are really, really, really large in size and shape. Whereas someone that has very low body fat, low fat content for their body, they actually have these cells. It's just uniformly speaking, many of them are smaller because they're not hoarding a large amount of fat within them. You're going to find fat almost anywhere we need to kind of like plug up space. So where we see kind of the heart tissue kind of fold in a little bit to give us the definition of where the atria and the ventricles are formed, fat will kind of take up space in those crevices. Again, in the stomach and the small intestines, you're going to see that you want the tube as it's kinked, as it's kind of like a rope of material kind of pushed into a small space. Fat will kind of help fit into those little crevices, those little canals, those little ditches to kind of help keep things in their spot and just keep them from having openings that, you know, tubes would fall or kink into. Right? So they take up a little bit of the extra space. They take up a little bit of cushioning ability to help things kind of sit better on top and stacked into each other, and then they store excess fuel. Okay? Reticular tissue is going to be the most open framework, the most spacious framework, and so this is going to be primarily seen in the liver and the spleen, where we want large numbers of cell-moving fluids to flow, but we want the resident cells to kind of have, think of like a bar. You want a few tables so people know like they can kind of congregate in an open space, and so your middle of your big, you know, bar vicinity has people, and people aren't like congregating to the walls. Okay, so the chairs, the stools, the tables that you place in your business in the middle of the room is kind of like the reticular fibers. They provide a little bit of a place for people to come dock and hang out, so they're not hanging out on the margins, on the walls, and nobody's in the center of the organ or center of the room. Okay. For your dense connective tissues, we're going to see less open space in the matrix. We're going to see less of the fluidy background, and we're going to see more richly protein content lined up side by side, and the cells are going to get squeezed a bit more in between the proteins. We're not going to maybe notice the cells as much, and that's one of the challenges of why sometimes being able to correctly identify dense connective tissue versus smooth muscle becomes challenging, because you don't necessarily see the cell nuclei, and so the proteins can be sometimes, depending on the stain and how they color, start to look like long skinny cells, but you don't really see their nuclei. Okay? So how do you name dense connective tissue? Well, if the main protein is collagen, all right, and it's a strong background matrix, it's going to be either dense regular connective tissue, because the collagen is basically uniformly lined up to be able to handle stress in a uniform manner, or the collagen is kind of a haphazard mess and it's irregular. If the main protein is elastin, it's elastic connective tissue. So looking again at the pictures of the book, you know this is a regular dense because you don't see a lot of open space and you see a lot of proteins, a lot of thick proteins lined up. All right. Now, the fibroblasts, the macrophages, the resident cells are embedded and kind of stuck in between the collagen. So you know that there are cells embedded in here, but most of the material that is the background material is a soluble, kind of suspended, tough protein that is collagen. The collagen is mostly arranged in a similar manner, running maybe longwise or lengthwise. And so this tissue does a very good job at handling, stretch or pull when you apply it from the direction that the fibers run. Okay? If you tried to pull on it in like the side direction, it's not as strong of a tendon or as strong as a ligament. So that's part of the reason why for ligaments and tendons, when you ask the joint to work in the direction it's supposed to, they are very good at what they do. But when you get the joint to be put in a position that it's asked to take a force in an abnormal way, so when you ask a knee to, plant and then twist, the ligaments and tendons around the knee are good at helping the knee plant and bend, but they're not good at twisting. And so that's why in many cases the knee ligaments can be torn when a knee gets twisted and not necessarily asked to bend or straighten. Okay? So most of your dense regular connective tissue are going to be those tendons and ligaments, and another form of a tendon is when it connects muscle to muscle. So an aponeurosis is what you call it technically when it connects muscle to muscle. So lots of fibers, a few cells. You still may have inflammation here. So when tendons and ligaments get damaged, the immune cells that live in that tissue are going to release inflammatory molecules that make it swell, that make it tender, that make it, not as usable as it should be, and that's what happens when you sprain your ankle or when you partially tear ligaments and tendons. And they take long to heal because if there's a lot of collagen here, there's not a lot of space for blood vessels to come in. And so it is hard for these cells to repair themselves because it takes energy to make proteins and energy to push them out, and energy requires oxygen and fuel. And if you don't have a lot of blood vessels readily bringing that fuel to these cells, then they can't produce the collagen to replenish what's been damaged or lost as quickly as they would if the blood vessel was sitting right there bringing a constant supply. Okay? For dense irregular connective tissue, you are going to see that it's still very collagen rich, all right? And this picture is showing blue, but it's just because it's a different stain. Technically, collagen, if you go through the same procedures, would stain pink like it did in the previous picture, but it's a different stain. That's the only reason why it turns out a little pinkish purple. So when you look at this picture, these collagen fibers are coming out at us. So imagine like the collagen fiber is running into and out of your computer screen, whereas this collagen fiber is running the long way. So you see its length. You don't see it in cross-section. You see it in laying down in front of you. Same with these over here. Now, the advantage to this, and you see it in the hypodermis, dermis region, you see it around cartilage and around bone is that it gives the tissue an ability to be pulled in this direction, pushed in this or that. So it gives the tissue an ability to handle stresses in a variety of directions and deal with those variety of directions in an ability to, counteract them. So these tissues, can handle being pushed and pulled, slide, squeezed, twisted, and a variety of stresses. All right? But the thing about these tissues is because it's not uniform, they're not quite as strong. So if they are damaged, they are like other tissues. It's dense. There's not a lot of capillaries. They are going to take slow time to heal, and they are going to be, when they heal, less strong. Okay, so again, you're going to find most of your capsules, most of your fibrous outer linings of organs might include a dense collagen-rich area, and so we're looking at dense irregular connective tissue. For elastic tissue, you're going to see that instead of the tissue being collagen-rich, there's more elastin fibers present. So not every ligament and not every tendon is just collagen. Some of them are going to have a higher amount of elastin protein. So the fibroblasts lay down collagen, yes, but they lay down a higher content of elastin proteins. And by having more elastin proteins, it makes this tissue more bendable, more stretchable. So elastin is going to be very important in some of the ligaments in and around our spine that are asked to kind of bend and pull and twist and handle more stress than some of the ligaments in our other joints that are more fixed joints that don't have as much freedom of movement. The heart needs a lot of elastic tissue because the heart, as it fills with blood, has to stretch. So you want what's known as the cardiac skeleton to be a collagen for strength, but elastin for stretch material. Okay? So you're going to see that this elastic connective tissue proper, it can be in elastic ligaments, can be part of different kind of areas around blood vessels that need to be stretchable vessels that need to be able to dilate pretty bigly, and like in the heart, where chambers need to be able to stretch. Okay? Moving to our specialized connective tissues, the first category being the fluid connective tissues, we are going to see now the matrix is not about collagen, it's not about elastin, it's not about fibroblasts, it's not about any of those things that were in the connective tissue proper. The matrix here for fluid connective tissue is water. And in that water, proteins are present, but the proteins are going to be hormones. The proteins are going to be transporting proteins. The proteins might be antibodies, part of the immune function. And so we won't necessarily see the proteins because they're dissolved. Now, the cells here are going to be some of our white blood cells, some of our immune cells, but majority, 99% of the cells are actually red blood cells. The red blood cells that are playing a role in what blood does, which is transport oxygen and transport that critical gas to cells so energy can be made. Lymph comes from blood. So lymph is going to be, like blood, the same background watery material with dissolved proteins, but lymph will have no red blood cells. White blood cells could be in lymph, and as lymph flows through lymph nodes, that is going to be a specialized reticular kind of capsule organ where lots of white blood cells congregate. So as the fluidy lymph moves through it, those cells can try to find anything in that fluid that shouldn't be there. So it's kind of like when water flows through a stream. Every now and then you want the water to flow through some type of filter or some type of netting. So if something is in the water that shouldn't be there, we capture, collect it, and can remove it. All right? And so think about the filters in your house. The water that comes from the ground outside goes through a few pipes, and in those pipes there's a few filters that then make sure some things are not then going to flow and still dissolved in the water that exits the faucet that you put in your glass and then drink through your mouth. Okay? All right. For blood, all of the background matrix is known specifically as the plasma, and all of the cells suspended in the plasma are known as the formed elements. Now, the red blood cell is 99% of the cells suspended in the blood. White blood cells make up the smaller next tier, and cells that break apart into cellular fragments are going to then be known as the platelets, those cellular fragments, and they're not quite dissolvable. They're not quite things that will go into the solution and disappear. You will see some of these with the naked eye. And platelets, again, are there for clotting and for the ability to plug injury holes and rips and tears in the chambers or in the vessel integrity of the walls. Okay. Again, lymphatic fluid comes from blood. This is known as interstitial fluid, and eventually interstitial fluid collects into a second series of pipes that is going to pass through lymphatic nodules where all these white blood cells congregate. And again, it's where we can filter the bodies of our, because we're 70% water, so we want to filter that water and make sure anything residing in that water shouldn't be there is able to be pulled out, destroyed, and taken out of circulation. Okay, so no red blood cells here. We will see all of the white blood cells like we saw in blood. We won't see platelets either, but all of the white blood cells, and again, in the lymphatic nodules, the cells are going to make a very thick kind of meshwork. So as the fluid moves through, kind of like flowing through a coffee filter, those cells are going to be able to interact with everything suspended in the fluid and pull out anything that shouldn't be there. Okay. The last category of connective tissues that are, again, specialized are our supporting connective tissue. Cartilage and bone specifically falls into this, and we know that cartilage and bone are going to make up our skeletal organ system. So in a way, the supporting connective tissues are the skeletal organ system. Cartilage is a little bit more bendable and pliable than bone. And the reason why it's more bendable and pliable is because the background matrix is more of a jelly, more of a Play-Doh-ish material, whereas the bone, it's hard, solid. All right? Now, bone has a little bit of bend because the hard, solid bone does have some collagen embedded in there. And collagen is strong, but collagen is a protein. And just like a chain link fence, a chains have a little bit of bend when the collagen is bent in a certain manner, but they're still very strong, okay? So that's part of the reason why when you are moving your joints, your bones move and they don't break every time you ask them to, for the elbow, flex or extend, all right? The little bit of bend that bone has allows the pull of the bone to happen, okay? Now, cartilage is going to be, for some of our bones, the original material that the bones originate from. Cartilage is going to be the material that is located at many of the ends of the bones that start to then form joints that have some interplay or movement. Cartilage is going to be some areas like the ears and the nose where we want to maintain a more pliable, manipulative area to the framework. The epiglottis, the back of the throat, the voice box, all of that is we want to have a pliable material throughout our lifetime, but we want it to still be robustly strong, so we use cartilage in those areas. Right? So the cells suspended in cartilage are going to get the name chondrocytes. Chondrocytes are going to technically want to be separated from the matrix slightly. So they are going to make a little bit of a chamber, a fence that they surround themselves within, and that chamber that they are residing in is called a lacunae. Since many of the bone cells come from chondrocytes, come from cartilage, our bone cells, which are going to be called osteocytes, are going to also, in many cases, live in a lacunae, a chamber. And that becomes from, again, some of them in a previous few evolutions of life were chondrocytes. So the chondrocytes and the osteocytes for some bones are cells that were at one point chondrocytes and then transitioned and became chondrocytes. Okay? Now, for the types of cartilage you have in the cartilage matrix, one of the things we're going to notice is cartilage matrix is not going to have blood vessels. So blood vessels were allowed in connective tissue proper. They were allowed in the space created by fibers. They were allowed to bring blood vessels, bring capillaries to interact with the cells, the fibroblasts, the immune cells. And that's how the immune cells in many cases came into the connective tissue, was because they exited out of the blood at the capillaries that flowed into those areas. Cartilage does not have capillaries. So we have less immune cells living in and around cartilage because they're not being brought to that area by vessels, by capillaries. The vessels are going to not be in close proximity to a lot of the cells living inside the cartilage. So those cells, if they get damaged, if they get destroyed, if they go through a lot of stress, are going to have a problem getting fuel, getting oxygen, and getting rid of their waste products. So that's another reason why if you damage cartilage, it's slow to heal. All right, and the chondrocytes living in their lacunae, suspended in the matrix of the cartilage itself, are going to produce that jelly material, and they are going to be the cells that produce the proteins that live in that jelly material. And the proteins are going to be our old friends, collagen, elastin, and if there's a lot of collagen present, the cartilage is stronger than normal regular non-collagen-rich cartilage. If the cartilage is full of elastic protein, then it's a very elastic bended cartilage, okay? If it's kind of a good mix between collagen and elastin, we just call it hyaline cartilage. To keep blood vessels from flowing into the matrix, the chondrocytes, in addition to making the proteins that live in the matrix and making the matrix background material, is also going to produce a anti-vessel protein. So the vessels want to grow towards cells, but they are going to be prevented from growing into cells because the chondrocytes are going to make a protein that makes an anti-vessel growing factor. And that anti-vessel growing factor is called anti-angiogenesis factors. Okay. All right. So in the perichondrium, the cartilage is surrounded by some connective tissue proper, and by being bound to connective tissue proper, that's where capillaries can flow near cartilage, and that's how the cartilage can then connect to the other tissues that it's near, like bone, like muscle, like the skin, all right? So the cartilage, because it's avascular, is going to still need some blood vessels to come close to it. So the perichondrium, the wrapping of connective tissue proper type tissue around the specialized cartilage connective tissue, is where we're going to see your fibroblasts, your immune cells, you're going to see your blood vessels, and you're going to see the mix of collagen, elastin, and then that material is what kind of connects the cartilage to the rest of the other area of the body. Right? So cartilage, when we grow it, if we want to grow cartilage in a longitudinal manner, so if we want like our nose to get longer and bigger, the cartilage undergoes what's known as interstitial growth. And interstitial growth means cartilage cells, those chondrocytes living in their little chamber of lacunae, making the matrix around them, and the collagen and elastin content in that matrix are going to multiply. And as they multiply, they are going to grow, and they are going to form two cells through mitosis. And then those two cells through mitosis want to separate from each other, and as they separate, their lacunae separate, and they start to put matrix between them. And as they do that, the cartilage then starts to get longer, and more of the chondrocytes then are able to push away from each other and grow a longer cartilage area. All right. So if you were to have your ears, kind of your earlobes, if you want to pull them and make them longer, kind of sit towards your shoulders, you would want to pull on the cartilage and stress those cells to undergo mitosis and lay down more matrix. And in doing so, the cartilage of the ear would start to get longer in size. Okay. Now, if I want to make cartilage thicken up, so I don't necessarily want the cartilage of my meniscus or my discs between my vertebra to get longer, because I don't necessarily want to get that taller, or like with the two bones, I don't necessarily have the space for the cartilage to get longer. But I want the cartilage to stay pretty thick. That kind of growth is going to be appositional growth. And this is how we get thicker and not necessarily longer in length. And the appositional growth is going to be occurring at the juncture of where the chondrocytes nearest to the wrapping connective tissue perichondrium live. And so what happens is the chondrocytes nearest this perichondrium will undergo mitosis and will start to lay down more matrix, and then in a sense kind of push the perichondrium further out, making the cartilage thicker. Right? So when you want to make cartilage grow, you want to make the growth go in a long direction. So you want the cartilage to get longer and taller. That's interstitial growth, and it occurs with the cells pushing away from each other, making more matrix in between them as they divide. If you want to see a cartilage area get thicker, you'll want to see that the cells nearest the protective layer start to grow and make the perichondrium push further out, so there's more material encapsulated in that perichondrium. Okay? For cartilage categorizing, if the cartilage has a good mix of collagen, elastin fibers, if the cartilage needs to be a little strong and a little elastic, it needs to have a good mix of those two proteins and a good mix of jelly substance, it is a hyaline cartilage. This is the most common cartilage because it's what gives rise to cartilage that becomes bone. It's the cartilage between all of the bones where we expect to find bone-cartilage-bone interactions. So it's the most common cartilage, right? And when we want it to grow, to get longer as we grow as individuals, it undergoes interstitial growth, and that's how our growth plates end up, again, growing long femurs, long tibias, long humerus. If we want that cartilage to thicken up, it'll grow in width and make those bones, you know, somewhat help grow in the width appositional growth. Okay. For elastic cartilage, you are going to see it in some specific locations where we want cartilage, but we want it to be a very elastic, constant cartilage that is being able to stretch. So the epiglottis, the voice box, those areas in the larynx that play a role in you being able to swallow, you being able for the extent of your life talk, that's where you're going to find large amounts of elastic cartilage. All right? Fibrocartilage, you're going to find it where you have a lot of weight bearing and you need a lot of strength. And because you need a lot of strength, yes, the matrix is jelly, but there's a lot of collagen living. So the discs that are weight bearing in your spine, all of them have a lot of collagen, and that's what helps make the disc strong and handle the weight that you apply on them. The meniscus of your knee, the symphysis in your pubis, so they're weight bearing areas where you need a big thick padding, typically is going to be this fibro collagen rich cartilage. Okay. The last of our specialized connective tissue is going to be bone. It's also known as osseous tissue. Bone can be brought about because cartilage changes to bone. And then another form of getting bone is dermal, irregular, dense connective tissue. So think about when babies are born, the fontanelles, that's not cartilage. That's kind of a dense irregular connective tissue like a ligament. It will kind of convert to bone. Bone cells are known as osteocytes. So osteocytes can come from mesenchymal cells. So that's why sometimes when people get tumors removed, when they look at what's inside a tumor, if the tumor had some connective tissue that started inappropriately dividing, some of those mesenchymal cells might have ossified. And so in my big fat Greek wedding, when one of the ladies talks about her biopsy of her little lymph or her little nodule, there was hair, teeth, and bone. Chances are it wasn't her twin. It was actually probably some mesenchymal cells in the cancer-forming process becoming bone fragments. Okay. Bone is going to be a calcium crystallized material with collagen fibers intermixed. All right, and we're going to go through a little bit more of this in the next in the 2 chapters when we get to skeletal tissue. But like connective tissue of chondrocytes or cartilage, the outermost lining of bone is a so around the bone osteum, and that is a regular connective tissue that has collagen elastin mixed. It's a dense irregular connective tissue, and the goal is to kind of let the calcium crystallized parts be a little separated from the muscle, from the ligaments, and so when they were pulled upon, when bone is pulled upon by ligaments and tendons, it's technically being pulled at by the periosteum. Okay. Again, bone, we categorize it as there are bones that is very dense, very compact, so there's a lot of calcium crystallized material and collagen mixed and pressed in together, whereas other bones we need them to be more light because maybe they need to have bone marrow embedded within. They need to be, you know, like the skull. We need to have air holes and passageways for things to flow. So that kind of bone is going to be a more spongy bone, a more porous bone. The material that runs and forms the struts and ruts is still crystallized calcium and collagen with osteocytes, but whether or not it's in a more kind of formal organized manner of dense bone or a more loosey-goosey manner of spongy bone and trabeculae will kind of tell us what's going on with the anatomical microscopic features of the matrix. Okay. So because some of the bone comes from chondrocytes, lacunae are still present. The bone cells are trapped in this matrix and they can't move. Unlike cartilage, blood vessels will come into bone, and part of the reason why we have open space in bone is so those blood vessels can penetrate the center or penetrate in between and get to the center of our bones. And so we're going to need little kind of gaps, little kind of canals for the bone cells to extend little like finger-like projections, little like arms towards the blood supply. And those little like ditches and canals are known as canaliculi. Okay. So when you look at most of your dense, compact bone, you're going to see these like logs running at you, and they have these little circular features, and all the little dark spots are osteocytes. There's blood vessels running in the center. There's blood vessels running in between, eventually radiating into the periosteum where they come from then bigger vessels that are connected to the bigger, you know, the aorta and the bigger blood vessels. All of these little osteocytes want to get close to those smaller vessels. So you see how they have like these little dark lines that are running towards these blood vessel rich areas. Those little indentation and ditches built into the matrix are called the canaliculi, and the osteocyte, kind of like a little daddy long legs, has these little arm-like extensions that are sitting in the canaliculi and reaching towards the blood supply. Okay? What do you call the individual rings or the little matrix material that's kind of taking up space around the logs? That is lamellae. So lamellae is the mix of calcium crystallized material and collagen fibers that are forming a solid matrix. Okay? If they are solid, forming a ring around a central kind of hole, they are known as concentric. They make a circle. If they are matrix that kind of between the little logs takes up the little bit of space, like right here, kind of like right here between these, you know, this log, and this log, they're known as interstitial lamellae. Near the outside of a bone, you'll find big circles running around and connected to the periosteum. Those are going to be kind of like the circumferential. So they are the circumference around the entire inside dense bone. Okay. The last part of connective tissue is this little bit of material where epithelial and connective tissue form these membranes. So one of the things about a membrane is it's two tissues, okay? It's a piece of connective tissue and a piece of epithelial tissue. And they form a kind of an organ, but it's not necessarily a large organ that falls in one of our organ systems. But it's two tissues that come together, and they are going to form what we know as some linings and some cavities. All right, so our pleural cavity, our pericardial cavity, our periosteum is all from these membranes. Synovial membranes are going to form a little bit of a cavity, a space that fluid will then enter into around where two joints or two bones come together at a joint. Mucous membranes, again, think about the air sacs in some of the bones of your face. They are going to be lined with mucous membranes that are going to, in most cases, have air in there, but when you're sick and the mucous membrane is overproducing mucus, can be full of gunk. Okay, so membranes play a role in lining and kind of covering and taking up some of these spaces that we have as the connective tissues are around or coming together as organs. They are usually formed by an epithelial tissue and its underlying connective tissue. All right. Now, typically the epithelial tissue is the one that's going to make whatever is put into the open space. So the epithelial tissue is going to play a role in what the membrane ends up being called. Most of the time the connective tissue is just the lamina propria, the background material where blood vessels, fibroblasts, and immune cells can sit and hang out behind the epithelium. All right, so our first type of membrane is a mucous membrane. So again, you have mucous membranes in your sinuses and all of those like air packets that we have in some of our bones of our face. All right, and they are there to help in some ways keep the bone with this open space light, but keep it to where if a pathogen is brought into the air that lives in that space, the pathogen won't get into the bloodstream and take over the body. So the epithelium protects that pathogen by trying to keep it outside of the body. Some of what it makes is a slimy type secretion, so that way that the pathogen might be trapped and might be captured. And again, we know with mucus that we blow out of our nose when we're sick, if it's overproduced because there's a lot of pathogens and these cells get overactive, the mucus will get very thick, will get very slimy, will get very green because it's full of pathogens. If it's loosey-goosey, white, and slimy, it might just be overproduced but not necessarily collecting and dehydrating. Okay? The connective tissue is typically going to be areolar connective tissue, so it's lamina propria, its background material that connects it to the underlying part of the body for the epithelium is going to be areolar tissue. The material that is mucus, the sliminess, not every single columnar or cuboidal cell of the mucous membrane is going to produce that material. Specialized glandular cells known as goblet cells, so they are going to be these columnar cells that kind of differentiate from the rest of the columnar cells or the rest of the cuboidal cells, and instead of just being there to kind of sit and be a barrier, they are going to be actively, constantly producing the mucus, and that mucus will then be excised out into the space and sit on top of the other cells. Those are going to be goblet cells. Okay? For our serous membrane, we don't really see the goblet cells. We just see the simple squamous epithelium. The simple squamous epithelium are going to make an ability or have an ability to put a few proteins and ions outside of the cell, and as they do, water will follow, and they'll make a little bit of a serous fluid, and it's sometimes called a transduate. This is going to be in many cases for the parietal and visceral pericardium, the parietal and visceral peritoneum, and the parietal and visceral pleural membranes, the simple squamous epithelial layer will be called the mesothelium. The lamina propria, like in the mucous membrane, will typically be an areolar connective tissue, so a mix of collagen and elastin, a mix of fibroblast immune cells, and capillaries living in that little area. Okay, and so again, we've gone through in Chapter 1, so this should be somewhat review that these cavities are formed because these serous membranes create a little bit of space and then put a little fluid in between it, so the lungs are technically separated from the heart is somewhat separated from the bones in the lungs, and the different areas of the GI tract are able to kind of fold and move and have movement, but it doesn't necessarily mean the other organs are affected. Okay? The cutaneous membrane is your skin. So this is a unique one in that the epidermis, the epithelial tissue, is a stratified squamous epithelium. The dermis is known as the cutaneous, or the dermis and the epidermis together form a cutaneous membrane. So it's one epithelial layer, one connective tissue layer. Here, the dermis, it's large like the epidermis, and so the area closest to the epithelial cells is an areolar connective tissue, but as you move deeper, the connective tissue becomes more dense, irregular. All right? So when you think about like cutting the skin and like, you know, pulling it off or tearing your skin, in many cases, if you see a little bit of fat and then a little bit of muscle underneath, you've torn the cutaneous membrane that's covering your body. Right? So there's really only one example of the cutaneous membrane, and it's the skin. The fourth type of membrane is at your joints that are going to have a lot of freedom of movement, and because of that and the way the joints the bones interact, there's a lot of space and crevices and bends. You want to fill that space with fluid, and so these membranes are going to be simple squamous epithelium and some areolar connective tissue and a little bit of a connective tissue capsule, so dense irregular connective tissue, and they're going to form a little bit of connections that are independent of the ligaments and the tendons that are running across the joint, and then they are going to help kind of create fluid that can be encapsulated where the bones are not necessarily touching and cartilage isn't located. Right? And so synovial fluid is produced by the epithelial cells. Okay. This is one membrane where when you look at it, there might be gaps between the tissue, the cells of the epithelium. So they're not always cells touching. It's not a perfect barrier. And so because of that, sometimes white blood cells that are in the connective tissue coming from the blood in the connective tissue might get into synovial fluid. So when people have swelling in their joints and they drain the fluid, they're draining some of that synovial fluid. There might be white blood cells if there's damage to the area. Okay. So where do all these connective tissue membranes and linings kind of come? Again, much of the connective tissue of the body is going to be found kind of connecting other tissues where they're lying. So the bone and the muscle are technically connected to each other by connective tissues. The bone is not necessarily touching muscle because around the muscle is connective tissue and around the bone is connective tissue. And when we start looking at our skin is not technically touching the bone and not technically touching the muscle because there's connective tissue. And when you start looking at the categories of what connective tissue is most superficial, collectively all of that superficial connective tissue is known as superficial fascia. So the hypodermis, which is the subcutaneous fat layer of adipocytes and areolar and reticular connective tissue together between the skin and the muscle itself, that is your superficial fascia. So you can call it collectively from the top of your head all the way to the toes. It might change in its fat content, change in its collagen elastin and its areolar and reticular content, but collectively all of that connective tissue that is made up of those three tissues is your superficial fascia. Right? When you go into the deep fascia, you're beginning to get underneath the muscle layer and maybe how the muscle and some of the space of the bone and muscle interacting, there's going to be more robust connective tissue and more fat located there that's going to be deep fascia. And at the Olympics, they were doing deep fascia massages and cupping. What they were doing with that cupping technique was trying to generate bruising, generate blood vessels to burst in this deep fascia, and by bursting the blood vessels, more immune cells ended up in this area, more repair happened in this area, so you were getting more blood supply to the connective tissue around the muscles. And the thought is you get more blood to the muscles, then they're bringing more nutrients to recover. Okay? The deepest connective tissue in and around your visceral, in and around your organs located inside the tissues and the cavities of the body will be your subserosa fascia. And so this is all of those serosal membranes that we talked about already. So it's just another way to talk about, again, the connective tissues and how they're organized in the body. If you go by anatomical organization, the connective tissues located closest to the surface of the skin are going to be superficial connective tissue, superficial fascia. The ones around the bones and muscles that form a little bit of that framework and assist in that are going to be your deep fascia, and then the connective tissues commonly associated in and around the organs in the body cavities, they are your subserosa fascia. Okay. So I know this got long again. I'm sorry about that, but a lot going on with connective tissue, a lot going on with epithelial tissue. So I'm going to do one more video to finish muscle and nervous tissue, and hopefully it'll be shorter. So again, this is all related to Chapter 4 and important for the week 2, week 3 labs that we do and the histology that we're going to ask you to look at in the microscope. All right, see you next recording.